Okay, so my talk today is about the prefrontal cortex, and I'm going to be trying to explain some of the ways in which neural processes that occur in the frontal lobe enable us to make decisions. So my focus is going to be on the region that you can see here. So it's the ventromedial prefrontal cortex, and this is bold activity in the ventromedial prefrontal cortex of the human brain that's occurring when a decision is made. And what I'm going to try and explain first of all is why we think this particular pattern of activity is so important for making decisions. So I'm going to explain to you how it is that we recorded this activity. So as I mentioned, we had human subjects. They were tested by Erie Borman as part of his doctorate a few years ago. And what he did was to ask them to make decisions between two options that looked a bit like two cards either side of a screen. So the subject's job was to pick between these two cards in order to earn points that at the end of the experiment were going to be translated into money. So the idea here is that the card on the left has 37 points, the card on the right has 55 points. So it might seem like the obvious choice is to pick the option on the right-hand side, but there's a bit of a catch. So in this particular version of the experiment, as we move through the experiment, through the 120 trials or so of the experiment, you can see that the probability that that choosing the card on the left, shown here in purple, or the card on the right, shown here in black, will yield the points that are associated with it, is gradually drifting and changing over time. So the subjects have to combine these two different pieces of information, the number of points they see on the card, that changes from trial to trial, with these drifting probabilities, to work out what's the best option to take on any trial. So let's imagine we're on the 42nd trial of the experiment, so round about here. Then the right-hand option is associated with more points, but the left-hand option is associated with a higher probability of winning. So if the subject factors both of those pieces of information into mind, then they might calculate the probability times the reward magnitude and come up with the idea that perhaps the expected value, when taking into account both the probability and magnitude, that it's better to take the option on the right-hand side. So what we did was to record this pattern of activity during decisions like these. And what we did was to calculate a critical variable on every trial of the experiment. We call it the decision variable. And it's how much better is the choice that the subjects took than the choice that the subjects rejected on each trial of the experiment. So let's imagine our 120 trials in the experiment, and what you can see here is that on each trial this number is going to go up and down to some degree, and what we're going to do is we're going to regress all of the brain activity that we see in each voxel of our MRI scan onto this series of numbers that we've recorded that report how much better the value of one choice is than another, the key decision variable for making a choice in this experiment. And if we do that, that's how we end up with this pattern of activity here. In other words, this activity here is going up and down in tandem with this decision variable here from trial to trial. Now we can unpack it a bit and see how it's varying over time. So at this point here, the information about the options is presented to the subjects. At this point here, the subjects are making their responses, and at this point here, they're finding out whether or not they've won the option on that particular trial, because remember, it's just probabilistic. And so what you can see in blue here is the bold activity, the blood oxygen level dependent activity that we can record with Fmri. And what we're looking at here are regression coefficients relating the bold activity to this decision variable. In other words, we're seeing how much the blood oxygenation levels are varying as a function of this decision variable on each trial. So what you can see is that the activity takes a few seconds to ramp up. That's because of the intrinsic delays that are associated with the blood oxygenation level response. But if we take those into account, what we can see, we know this normally has a delay of several seconds, but if we take that into account, then we can imagine that we can infer that the neural activity that caused this signal must have been occurring just before the time when the response was made. We can unpack the activity even further and decompose it into two different parts. So a part of the variation in the bold signal is related to the value of the choice that the subjects take on each trial, and another part is related to the value of the choice that the subjects are rejecting on each trial. So just to summarise then, we think this activity is important for decision making because it's related to the value of a choice that you take and the value of a choice that you reject. And because of a number of other experiments, we think that this is what we have to do at the moment when we're making a comparison between one option and another in order to make a decision. So far then, I've been concentrating on this region of the human brain here. And what I want to move to now is the species that I'll be focusing on for quite a bit of the talk. So this is the macaque monkey. And what we can see here is a different type of view through the macaque monkey brain. So now here we're looking at a coronal cross-section, so a cut through the brain like that, rather than a sagittal section, a cut through the brain like that, that we're seeing for the human brain over here. And it's highlighting this part of the monkey brain, the orbitofrontal cortex. So this is a region that's very close to this medial frontal area here. And it's a region where people have found, when they've used microelectrodes to record activity, that there are neurons that have activity that is related to the value of choices that the monkey is considering. And what I want to talk about today is the question of whether or not this brain region here that's been highlighted in these monkey studies is the same one as this region here that's been highlighted in these studies of the human brain. Now, for many years we've thought these regions are quite close together, so perhaps what we learn about the neurons here in this region of the monkey brain have some bearing on our understanding of what this region in the ventromedial prefrontal cortex is doing. But if we look closely at the anatomy of these regions, it actually turns out that they're probably not one and the same region. This ventromedial region of the human brain is actually probably most similar to the ventromedial part of the monkey brain. We can demonstrate this by looking at patterns of Fmri activity in the monkey brain and in the human brain. So one way that we can do this is we can take the bold signal in this brain region here while our human subjects are at rest, and we can look across all of the other areas of the brain to see where it's positively and where it's negatively correlated. And in fact, if we look at the set of areas where there is either positive or negative correlation in the human brain, that sort of constitutes almost like a fingerprint, an identifying signature for this human ventromedial prefrontal area. And so what we can then do is go and have a look at the macaque monkey brain, recorded with Fmri while the animals are just at rest, to look to see if we can find an area with a similar type of fingerprint. So with the same patterns of positive and negative correlations with other areas in the macaque monkey brain. And if we do that, so just to remind you, what we're doing is we're recording the signal from this region here in blue, and then we're going to compare it with the signals from multiple other regions in the macaque monkey brain. And if we do that, then what we can find is that this ventromedial region in the human brain has positive correlations with some areas and negative correlations with other areas. So you can see that every place where this line is close to the periphery of this circle, this ventromedial prefrontal area, has a strong positive interaction with these other brain areas. Wherever the line is far from the circumference of this circle, then there is no interaction or even a negative relationship between the brain signals. And so what we've got in the two different colours here are the pattern of interrelationship between that ventromedial prefrontal signal in the human brain, and then also in the other colour, in the macaque monkey brain. And in fact, what you can see is that the patterns are quite similar if we look at this region here, the macaque monkey brain. This region here is a very similar correlation pattern with the rest of the macaque monkey brain as this human region here has with the rest of the human brain. And in fact, what's shown in this picture here is a heat map where the brightest, most orange-red voxels in this monkey's brain are the ones that have the closest correspondence in terms of their interaction with the rest of the brain, as does this ventromedial prefrontal region in the human brain here. So the brightest, most red areas are most similar to this human ventromedial area. So that's telling us then that this ventromedial area is in fact most similar in the monkey to this ventromedial area in the human brain. So this is important because we know that this ventromedial area of the monkey brain is quite different from the orbitofrontal cortex in many respects. So it's adjacent, but it has very different anatomical connections. And this is summarized on this slide here. So what you can see is a set of anatomical results that are produced by injecting a tracer into the medial frontal cortex in this region here, in the region highlighted in blue, or in this region here, highlighted in red. And then all of the other blue and red dots show you where the tracer is going to in the macaque monkey brain. And what you can see is that after injections in the medial frontal cortex here, we get lots of labeling through other parts of the medial frontal cortex and in the entorhinal cortex here. But there is almost no overlap with the areas that are labelled by injections in the orbitofrontal cortex here. So you can see there in the perirhinal cortex and in the STS, for example, and other parts of the orbital surface. There are just one or two small areas where the dots overlap. So what this is telling us then is that the orbitofrontal cortex, the region in red here, is in receipt of very different types of inputs and exerting very different types of influences to this region in blue here, the medial frontal cortex. They're quite distinct areas of the brain. So perhaps then the patterns of activity that we've been recording in single neuron recording studies in the orbitofrontal cortex doesn't tell us directly about this medial frontal area in the human brain. And perhaps we've been conducting rather different types of experiments when we look at monkeys and when we look at humans. So if we take an example of an experiment where macaque monkeys have learned about the values of different stimuli. We're going to look at this one here from Steve Kennelly, where monkeys were taught about many different stimuli that had different probabilities of reward, or different amounts of reward, or different costs associated with them. And in this particular case, the animals have learnt many different stimuli, but they've learnt them over an extended period of time. Typically in these experiments, the animals spend several days or weeks or learning these stimuli. But this is quite different from the way in which we test human subjects in these types of experiments. So, for example, in experiments on human subjects, for example, in this experiment by Chow and colleagues, typically what we find is that we're teaching them certain rules to use for selecting the best option. So in this particular case, from Bolt and Chow, subjects were taught about the orientations of stimuli and their colours. And the orientations and colours indicated different probabilities or magnitudes of reward. And typically in these types of experiments, the subjects are given some brief instructions, a little bit of practice, perhaps just for half an hour or so, and then they go straight into performing the task. And sometimes they might end up making decisions about options, about stimuli that they're encountering for the very first time during the course of the experiment, but they're using the rule that they've learnt earlier in order to make an inference about the value of this particular stimulus that they're encountering now. So it's quite a different way to do the task in comparison to the macaque monkeys. Instead of learning things in a very hard, fixed way, the human subjects are actually making new inferences about the values of these options that they're encountering based on the previous experiences that they've had in the past. And this is actually something that we often do. So, for example, if you're thinking about the next foreign holiday destination, when you're next able to travel abroad, if, for example, you are considering different holiday destinations in Italy, for example, such as Florence and Syracuse, then you might be able to make that decision even though you've never visited either of those locations in the past. But you can come to some sense of what might be the best location for you to choose on the basis of considering different aspects of both of these destinations. So, for example, you might consider the possibilities for sunbathing or swimming in these different locations, or the possibilities for visiting cultural sites, art galleries, and museums. And you might be able to consider these options on multiple different dimensions and make an inference about which would be the better destination for you, given the way in which you think about these different dimensions that define each of these locations as a holiday destination for you. So that's essentially what the human subjects are doing in the experiment by Chow and colleagues, but it's not really what animals are doing in many animal models of value-guided decision-making. So what I'm going to tell you about is an experiment conducted by Alessandro Bongiorni with lots of help from Miriam Klein-Pfluger. And this is an experiment that was designed to try and investigate this mode of decision-making, which seems very natural to us when we're making our holiday choices, and very natural for us when we're designing experiments for human subjects to perform. But it seems to be more difficult to think about how we might train animals to perform decisions in this way. But in this experiment, we've tried to get around some of these problems. So we've tried to create a situation where animals are learning about information and using it to perform decisions in a manner that's a bit more similar to the way in which humans make value-guided decisions. So what Alessandra did here was to teach animals about different colours that were associated with different magnitudes of reward. So in this particular case, the different colours predict different numbers of drops of juice, of a fruit juice that the animals like eating. Then Alessandra taught the animals about a different aspect of the stimulus. So he taught them about the number of dots that were shown on the stimuli. And this provided information about the probability that the stimuli would yield a reward. One of the important things that Alessandro did when he taught the animals about these two sets of stimuli was that when he taught them about the first set, he always kept the probability of reward constant at about 60%. And when he taught them about the second set, where the probability varied, the magnitude of reward, the number of drops of juice that were being delivered, was kept constant at about 5 drops of juice. So what you can see then is that Alessandro taught his animals about two stimulus dimensions, one that was associated with variation in probability and one that was associated with variation in magnitude. And importantly, what you can see is that having taught this, Alessandro has taught his animals about a two-dimensional space of choice options that they can. So he's done this by training his animals just on a very limited part of this space, so that they've just had experience of all of these different reward magnitudes at the 60% level, and they've had experience of all of these different reward probabilities at this 5-drop reward magnitude. But nevertheless, in conjunction, these magnitudes and probabilities define a two-dimensional decision space. So that means that having trained the animals on just these two paths through the decision space, Alessandro could give his animals novel stimuli that they hadn't encountered before, and he could test to see whether or not they were able to make inferences about the values of these novel stimuli on the basis of what they knew already about these probabilities and magnitudes. So, for example, Alessandro could give his animals a choice between this familiar stimulus here and this familiar stimulus here, F1 and F2, or he could give them a choice between these unfamiliar new stimuli, which have elements that are familiar, but they've never been encountered in this particular combination before in the past. So Alessandro gave his animals these new stimuli to choose between while they were actually in the MRI scanner and while fMRI data was being recorded from them. So in a sense, the animals are going to be in the type of situation that our human subjects are typically in when they're making value-guided decisions. They're actually making inferences on the fly about the values of these options on the basis of past information that they've stored from similar types of options. But they won't have encountered exactly the same stimulus features in the same combinations in the past. So one of the experiments I'm going to tell you about is going to involve making decisions in this way. The second experiment is going to be one where the monkeys just encounter one option at a time. And we're going to do this because it tells us, it provides a sort of routine for looking at the nature of the representation in the macaque monkey brain, the way in which these choice options are being represented in the brain. And then finally what we're going to do is we're going to look at what happens when we disrupt this brain area and see how it changes the way in which decisions are being made. So we're going to look then at a series of experiments. The first two experiments were collected on consecutive days that were actually interleaved with one another. There was then a 6-month gap while we were analysing some of the data. We then retrained the animals on the very over-familiar version of the task, and then gave them the novel options once again for experiment 3, where we were looking at the effect of disrupting neural activity. Okay, so we're going to start off then with this first experiment. Remember what we've got then is we've got all of these different options that the animals might experience in these different points in this two-dimensional value space defined by probability and magnitude dimensions. So just to point out, the familiar and the novel options that the animals might encounter are very similar. So the stimuli are going to look like this. They're coloured stimuli made up of different numbers of dots. So the colours, the reward magnitudes, the dots indicate the probabilities. And they can be, when they're familiar options or novel options, they can be either closer together or further apart in their value, and so that's going to make the decision easier or more difficult. And of course it's also going to mean that we've got a variable, a bit like the one that we had in the very first human neuroimaging experiment by Erie Borman that I told you about at the beginning of the talk. So we can look at the brain to look to see if we can find activity that is covarying with this decision variable, how much better one option is than the other option, and how that's being used to guide making a decision. One thing I should say is that even when the animals are encountering these novel options, they're still able to make correct choices on many occasions. So you can see here on the x-axis, we've got the difference in value between choices on the right-hand side or on the left-hand side. And then we're looking here at the proportion of occasions that choices are made to the right-hand side, or down here on the other end of the y-axis to the left-hand side. And we've got data from 4 individual monkeys, and you can see that one of them's a bit worse than the others. They're all reasonably good at making accurate decisions, even if they might not have encountered these precise stimulus conjunctions before. Okay, so now let's have a look at the brain activity, and we're going to look at the brain activity that's time-locked to the moment of making this decision, just as we did in the human neuroimaging experiment that I told you about earlier on. And if we look for activity that's related to the value comparison, how much better is one option than the other, then we can see that there is activity that's covarying with that number as it changes from one decision to the next, as we move from one trial to the next. So we can see this area here in the orbitofrontal cortex is one of the key places where the activity is changing in tandem, in line with that decision variable. So this is the orbitofrontal cortex region where many cells have been linked before to the encoding of choice value. However, this is the activity pattern that we see when we look at all of the trials together. When we just focus on the novel decisions and we look for this value comparison signal and look to see where is it stronger on novel trials as opposed to familiar trials, then we see that it's most prominently different here in the ventromedial prefrontal cortex of the macaque monkey. And again, in both cases, what we can do is we can zoom in and look at the activity pattern in this area. And you can see that there is activity that's associated with the unchosen value that's being rejected in green, and there is activity related to the chosen value, so the value of the option that's being selected. And you can see, while there's a very general signal here in the orbitofrontal cortex, there's a much more specific one in the ventromedial prefrontal cortex that's associated with making these novel choices using novel combinations of stimulus features that haven't previously been encountered. So it seems then that when we test our monkeys in a similar way to the way in which we test humans, and we start to see this value comparison signal here in the medial frontal cortex. Okay, so far then we've been focusing on the results from experiment 1, where the animals are comparing different choice options. Now let's have a look at experiment 2, where the animals encounter just one option at a time. So if you remember, these are from interleaved testing days, and the reason that we're going to look at just one stimulus at a time is because we know that in many areas of the brain, there's a phenomenon that occurs that is called repetition suppression. If the same stimulus is presented on 2 occasions, one time here and one time here, then the brain activity that's recorded on the second occasion is less than on the first occasion. It's as if, in some way, something about the using the same sets of neurons on 2 occasions in quick succession leads to adaptation and reduction in activity on that second occasion. So we can exploit this effect to tell us something about the nature of the representation of the choices in different brain areas. We know that if we change the second stimulus so that it's very different from the first stimulus, then the response, the neural activity, won't adapt. It will return to its original level. So we can exploit this fact and look to see how adaptation occurs as a function of different stimulus features that define the choices in these different brain areas. So this is what we've done here, and we're going to be focusing on results from the orbitofrontal cortex. And what I want to turn your attention to is this result here. This is actually telling us how much adaptation we're getting in that orbitofrontal region when the options that are being presented on the screen, one trial after the next, are completely different in value, as opposed to being identical in value. That's what this label is indicating here. It's a difference in activity for identical value as opposed to different value stimuli. And you can see when they're identical value stimuli as opposed to completely different, then we're getting lots of suppression in activity in the orbitofrontal cortex. By contrast, when we have stimuli that are the same, have the same probabilities but not magnitudes, or the same magnitudes but not probabilities, or when we have stimuli that are the same value but they have the same value as a result of different probability and magnitude components, then the degree of suppression is greater, but not significantly greater than in the completely different value case. In other words, it seems as if in the orbitofrontal cortex we get the most repetition suppression when we have an absolutely identical stimulus being presented on 2 consecutive occasions. It's as if the orbitofrontal neurons in this area are encoding the option in terms of all of the features of these stimuli, both the probabilities and the magnitudes. But it doesn't seem as if the ventromedial prefrontal cortex is doing the same thing. As you can see here, there's an absence of any repetition suppression effect like this in this medial frontal cortex region. So what's going on here? What type of neural representation is mediating the decision signals that we saw in the medial frontal cortex? Well, we can test for a different type of neural representation in the medial frontal cortex. If we think about the way in which stimuli differ from each other under a different framework, a different type of framework. An alternative way to think about how the stimuli differ from each other on successive occasions is in terms of their position in this 2-dimensional value space that we've looked at before, the probability of reward and the magnitude of reward. We can think about the way in which successive stimulus presentations from one trial to the next appear at different locations in this 2-dimensional space. And we can think about we can think about trajectories through this space that might occur each time a different stimulus is presented with respect to the previous stimulus on the previous trial. Now this might seem like a slightly odd thing to do, but in fact it's something that people have done regularly when they've been analysing brain data, albeit not so much when they look at decision-making activity, but when they look at spatial encoding. So let me explain this a bit more now. If we, what I want to turn to is an experiment that was conducted a few years ago that was designed to look at how spatial representations are built up in the brain. And in this experiment, they asked their participants to move around a virtual spatial arena. And they recorded the directions they moved through this spatial arena. And they did this because they knew that in a part of the brain, there is a type of cell called a grid cell that has these very unusual types of receptive fields. So the neurons become active when the individual is in a number of different locations that cover the whole of this arena. And one of the interesting things about the way in which these active locations are arranged is that they are perhaps most easily described as falling on a hexagonal grid, a grid that repeats every 60 degrees. So you can imagine that if you're moving along this direction here, or along this direction here, you're going to be moving across the space in a way that is going to activate this particular neuron very strongly because you're moving in parts of this neuron's response field that activate this neuron. And one of the interesting things about neurons in these parts of the brain is that all of them have their grids aligned to the same orientation as one another. So what this means is that whenever there is movement that is occurring in line with this grid, there is going to be lots of activity, even in the average signal that we get using Fmri. And there's going to be much less whenever they're moving in a direction that's misaligned with this grid. Then there's going to be more activity when they're at 60 degrees to that original direction, or 120 degrees, or 180 degrees with respect to that original direction, as opposed to all of the other intervening directions. So what this means is that the Fmri signal should go up and down depending on whether or not movement in this spatial arena is in line with this grid or at any of these 60-degree repeat patterns shown in red, as opposed to any of these other patterns that are offset from the 60-degree repeat pattern. So this pattern of activity is seen in the entorhinal cortex. This is in the human brain here, but it's also and it's known to be prominent in the activity of single neurons in this same region that are recorded in a variety of different animal species. But intriguingly, it's particularly prominent in the medial frontal cortex of the human brain. So what we can do then is we can test to see whether or not the medial frontal cortex of our macaque monkeys has a similar type of grid-like representation, not of a real space that we might move around in an arena, but of this more conceptual space or value space that is being used to guide decision-making. So whenever successive stimulus presentations are falling along in register with this grid-like representation, then there should be strong activation. But as the trajectory that is moved through this conceptual space falls further away from this grid orientation, then there should be less and less activity until we move to a position that is at 60 degrees from this original orientation. So what we can do then is we can look for evidence of this hexagonal modulation of activity in the macaque monkey brain to see whether it's occurring as a function of movements through this conceptual space, this decision space. So if we do that, then we find that indeed that's the case in this medial frontal area. So we can see that there is significant evidence for this hexagonal pattern of activity modulation, but there is no evidence from modulation with these other periodicities, 4-fold, 5-fold, 7-fold or 8-fold periodicities, as opposed to the 6-fold periodicity, the hexagonal pattern that is indicative of a grid cell representation. So this is telling us then that these macaque monkeys are perhaps navigating this novel decision space as a function of probabilities and magnitudes in order to decide what are the best novel choices to make. And we've got a very different type of representation of this decision space that can be used to make novel inferences about new choice options in a way that wasn't possible in the orbitofrontal cortex in the macaque monkeys. Okay, so far then, what I've showed you is that there are decision-making signals in the ventromedial prefrontal cortex, and then I've also shown you that there is evidence for a grid-like representation in the medial frontal cortex in the macaque monkeys. Now, what I'm going to show you next is what happens if we disrupt this activity pattern. And what we're going to do is we're going to have a look at the impact of a relatively new, minimally invasive disruption tool that we've been developing recently called Transcranial Ultrasound Stimulation. So transcranial ultrasound stimulation is something that a few of you will have probably come across, but there are probably many people who haven't heard about it. And this tool is designed to focus a beam of ultrasound onto the brain. And the important thing about ultrasound is that it can actually be focused at some depth from the transducer. So that means that we can target brain regions with the ultrasound that are relatively deep in the brain and far away from the surface. So this gives us a tool to stimulate deep in the brain and it's a type of tool that we don't have for looking at in a minimally invasive way that we haven't had before. For example, transcranial magnetic stimulation only allows us to look at relatively peripheral dorsal and lateral brain regions rather than deep in the brain. So I'm going to be telling you, first of all, a little bit about why we think this technique works, why we think we've got evidence that it really does stimulate and reach the brain. And I'm going to be telling you about some experiments conducted in Oxford by Davide Follony, Nenad Verhagen, and Jerome Selle. So here, what I'm going to be telling you about, first of all, are some experiments where either this brain region here, in the cingulate cortex, or this brain region here, in the amygdala, were targeted. And then what I'm going to show you are some recordings that we took from the brain, showing that the ultrasound targeting had an impact on brain activity. And the way we're going to measure that impact, the way we're going to record the neural activity, is using Fmri. Okay, so what we're going to do then is we're going to look at the impact that the ultrasound is having by looking at these fingerprint representations of the brain that I've shown you before. Here's one for the anterior cingulate cortex. As before, we've got all of these brain regions on a circle circumference here. And then if we focus first of all on this blue line, this is telling us how strongly the anterior cingulate cortex in a normal control situation is coupled with all of these different brain regions. Now the next thing that we can look at is shown here in red, and this is the case when we stimulate the anterior cingulate cortex with ultrasound. So these red data here are significantly different from the blue data. So in other words, the cingulate cortex's connectivity with the rest of the brain has been significantly reduced when the ultrasound has been directed to that singular area itself. In other words, something about the way in which we're putting an ultrasound into that cingulate region is stopping it from communicating in the normal way with its normal partners that constitute its connectivity fingerprint. Now, last of all, we can look at the yellow data here. And these data come from recordings taken from the cingulate cortex, but now the ultrasound is directed towards the amygdala. And now you can see in yellow that although there are some differences, there is no significant change in the overall fingerprint compared with the control state shown in blue. Now here's another data set, and this is in fact a complement to the one that we've just been looking at. Now we're looking at the amygdala brain region's fingerprint of connectivity with the rest of the brain. Here it is in blue in the control condition. Now in red, what we've got is the situation where we look at the amygdala's connectivity when the cingulate is stimulated, and you can see it's not significantly different. But now, in yellow, we've got the case for the amygdala's connectivity when the amygdala itself is stimulated. And now you can see there's a very stark, strong difference. So, this is telling us then that when we stimulate one of these areas, just that area's connections and interactions with the rest of the brain are being changed and disrupted. We can look at these data in a different way. We can map the region of connectivity change that's occurring when we stimulate the cingulate cortex. That's shown on these parts of the figure on the left here. And you can see the region of change is here in the cingulate cortex, in fact, slightly deeper than we'd intended. And that partly relates to the way in which the ultrasound is reflected by bone and other features of the skull. And again, here we've got related data for the amygdala. And again, in fact, the stimulation is perhaps slightly deeper than we'd intended, again, because of reflection processes that occur to the ultrasound. Okay, so that was just some evidence then that ultrasound can be used as a tool for stimulating the brain. What we're going to look at next is we're going to try and see what happens to behaviour when we stimulate this medial frontal cortex area. So we're going to look at novel decisions, and we're going to see whether or not we can change the way in which novel decisions are made when we beam ultrasound into the medial frontal cortex. Now, this figure here is to remind you that in order to make these decisions, the animals have to think about the probabilities and the magnitudes of the options they're considering. And if they multiply those magnitudes and probabilities together, or if they act as if they're multiplying them together, then they'll work out that stimuli down here have a very high value, that's shown in yellow, whereas stimuli up here, with low magnitudes and low probabilities, have low values over here. But on this part of the figure here, we can show that actually animals can approximate quite good choices if they don't really integrate the two different dimensions of the stimuli in the optimal manner. Even if they don't integrate them multiplicatively and they just add the different probabilities and magnitudes, or act as if they're adding them, then they still have an approximate sense of the values of the options. You can see it plotted here in the colours, and you can see that these colours here are similar to, but significantly different from these colours here. So what we can do then is we can look to see if we disrupt this two-dimensional grid-like representation of the value space that we know exists in the medial frontal cortex, do we change the way in which animals make decisions from this optimal pattern to this more additive approximation pattern? And we think that's the case. So the way in which we think animals typically make the decision is that to some extent they're acting as if they're multiplying these options, which is the optimal thing to do. But to some extent they're acting as if they're adding them together, they're acting as if they're approximating by just additively combining magnitude and probability. And we can express this combined approach in this way here. What we can think of the animals as doing is, to some degree, acting as if they're integrating the values multiplicatively, but to some extent as if they're just combining them additively. And this parameter here, beta, is our integration coefficient. So as it gets higher, the animals are multiplying them more, or acting as if they're multiplying them, and they're acting less as if they're adding them together. And importantly, by looking at the pattern of decisions that our monkeys make, we can infer whether or not they're acting in this more multiplicative or additive manner. So we can check whether or not we can recover these different potential values for this integration coefficient from the type of data that we're likely to record, and indeed we can do that. So what we can do then is look at our animals. We can try and calculate this integration coefficient, which, if it's higher, means that the animals are likely to be acting as if they're integrating the different component parts of the stimulus values, or if they're just more likely to be combining them additively, which is what would happen if you're just thinking about the different dimensions separately, but not combining them in the optimal way. So what you can see here is data for a controlled situation from 3 different individuals, and you can see that they don't perfectly integrate. This animal here is almost perfect. The other 2 are tending in that direction. Here we've got some data from actual stimulation of a control site, and here we've got data from this from near the centre of the activity region that we've identified with Fmri. And now what you can see is a significant decrease in that integration coefficient in the different animals that we find is reliable across days when we repeat the experiment again and again. So what we've seen there is that when we stimulate in this region, the animals still make decisions. They've still got some sense of the value, but it's as if they've got a sense of the different component parts of the value, rather than being able to combine those different component parts in the way that you would expect in a sort of 2-dimensional sense of the choice space. So just to summarise then, what I've shown you is that probably we use this brain region here, this medial frontal cortex region, to make novel types of decisions, perhaps when we're making inferences about values, rather than simply retrieving a memory of an experience of exactly the same stimulus that's been encountered in the past. Instead, we seem to be constructing the values of these options based on different elements of similarity that the options have with other stimuli that we've encountered in the past. And if we test humans and macaques in a similar way, allow them to make inferences about novel choice options, then we see that it's a similar brain region that's important for making the decisions in both cases, and disrupting it disrupts that decision-making pattern. Okay, so the final thing then is that I just wanted to say thanks to all of the people who've helped with collecting all of the data that were used in these experiments. And thanks very much for paying attention.